On this trip through the digestive system, we will study secretion, the production and release of juices and hormones by the GI tract and its accessory glands. Your goals for learning are to list the secretions found in each part of the GI tract, to associate each secretion with its functions, to explain the control of secretion in each part of the GI tract. Here's what you need to know. Each secretion of an organ is made by a specialized cell type. The anatomical organization of the autonomic nervous system. The histology of the alimentary tract wall. What enzymes are, how they work, and why they are needed for biochemical reactions. To see definitions of terms, click the bold red words. The GI tract is very busy after each meal. Its organs add a large volume of fluids to ingested food as it is moved along and digested. Click the mouth to see an average day's worth of food and secretions of the GI tract in a normal adult. Suppose you consume 800 grams of solid food and 2 liters of fluid in a day. About 1.5 liters of saliva are secreted into the mouth. Click the stomach. The stomach secretes 2 liters of gastric juice. Click the duodenum. The pancreas delivers 1.5 liters of fluid, and the liver delivers half a liter of bile into the duodenum. The small intestine secretes 1.5 liters of fluid. The total amount of fluid entering the GI tract from all sources is 9 liters. Click the terminal ileum. The small intestine absorbs 8.5 liters of fluids and most of the ingested food. Click the large intestine. The large intestine absorbs 0.35 liters of fluid. The numbers we have supplied are estimates, but they provide a guide for understanding the large volumes of fluid that the GI tract handles in a day. In summary, the GI tract receives 9 liters of fluid in a day and eliminates only 0.15 liters. Of the 800 grams of food ingested, only 50 grams of indigestible food are eliminated. Less than 10% of ingested food is eliminated under normal conditions. The salivary glands produce saliva. The periparotid, submandibular, and sublingual glands are called the extrinsic glands because they lie outside the oral cavity. There are smaller intrinsic salivary glands within the oral mucosa. The parotid glands contain only serous cells that produce a watery fluid containing enzymes, electrolytes, and a small amount of mucin. The other extrinsic glands contain both serous cells and mucus cells that produce a more viscous fluid. Saliva is produced in copious volumes and is delivered through ducts to the mouth. Its functions include protection, taste, lubrication, and digestion. Click Protection to learn more. The volume of saliva produced in the mouth can dilute hot drinks and foul-tasting substances and buffer acids. Saliva protects the oral mucosa, cleanses the mouth, and helps prevent dental caries. Saliva contains lysozyme, an enzyme that attacks bacterial cell walls, and IgA antibodies that are active against bacteria and viruses. Click Taste to learn more. Water is the primary component of saliva. It moistens food for swallowing and dissolves food molecules. The sensation of taste requires molecules to be in solution. Click Lubrication to learn more. Mucus lubricates food particles, making them easier to swallow. Click Digestion to learn more. Chemical digestion occurs in aqueous solutions. Saliva contains the digestive enzyme amylase that begins the digestion of starch in the mouth. Intrinsic salivary glands on the tongue release lingual lipase. It is optimally active at an acidic pH and may digest some triglycerides in the stomach. The control of salivation is unique in two ways. First, it is mediated almost entirely by the nervous system. Note that both gut hormones and neural reflexes regulate secretion in other portions of the GI tract. Secondly, both the parasympathetic and sympathetic branches of the autonomic nervous system stimulate salivation. Note that parasympathetic and sympathetic nerves have opposing actions on most other organs. Click the parasympathetic fibers of cranial nerve 7 or 9. The parasympathetic division of the autonomic nervous system is the primary controller of salivation. Parasympathetic fibers are carried in the facial and glossopharyngeal nerves, cranial nerves 7 and 9. Parasympathetic activity initiates and maintains salivation. In general, parasympathetic stimulation produces large amounts of watery saliva containing enzymes. Click the brainstem to reveal stimuli for salivation. The thought, smell, or taste of food stimulates the salivary center in the medulla to increase parasympathetic activity and salivation. Acidic substances like a piece of lemon and pressure of chewing food or non-food in the mouth, like a peach pit, are powerful stimuli for salivation. Intestinal irritation and accompanying nausea are also powerful stimulants of salivation. Click the cerebral cortex to reveal stimuli that inhibit salivation. Fear, fatigue, sleep, and dehydration all inhibit salivation. Click the sympathetic fibers arising from T1, T2, and T3 of the spinal cord. Activity in the sympathetic nervous system produces a small volume of saliva that is thick with mucus. Because sympathetic stimulation accompanies frightening or stressful situations, the mouth may feel dry at such times. In summary, both parasympathetic and sympathetic activity increase salivation. Both also stimulate metabolism and growth of the salivary glands. Mucus is the only secretion produced by the esophagus. Its function is lubrication. Click lubrication to learn more. Remember that the esophagus is simply a conduit between pharynx and stomach. It serves no digestive or absorptive functions. Since it is long and flattened when empty, keeping it lubricated with slippery mucus facilitates passage of a bolus along its length. The gastric mucosal epithelium is made entirely of secretory cells, including exocrine, endocrine, and paracrine cells. Exocrine cells secrete products into the stomach lumen. These products make up gastric juice, which includes mucus, pepsinogen, the precursor to the enzyme pepsin, hydrochloric acid, and intrinsic factor. Click each component of gastric juice to see where it is secreted. When you have finished, click Continue. Mucus is secreted throughout the stomach. Pepsinogen is secreted throughout the stomach. Acid and intrinsic factor are secreted together from the same cells in the fundus and body of the stomach. Enteroendocrine cells secrete gastrin, and paracrine cells secrete histamine into the interstitial space. Click the hormone and paracrine to see where each is secreted. Gastrin is secreted in the pyloric region of the stomach. Gastrin enters the bloodstream and passes through the circulatory system before returning to the stomach to exert its effects. Histamine, like hydrochloric acid, is secreted in the fundus and body of the stomach. Histamine is a local chemical messenger that acts on nearby cells. Remember, histamine is secreted into the lamina propria, not the stomach lumen. The surface of the stomach has deep wells called gastric pits. Each pit leads to gastric glands. Click the entrance to a gastric pit to see an image of a gastric pit and gland typical of the fundus and body regions of the stomach. Throughout the stomach, gastric pits and glands contain cells that produce mucus and pepsinogen. In the fundus and body, gastric glands are deep and contain cells that produce both acid and intrinsic factor and cells that produce histamine. Click each cell type to reveal its secretion, then click Continue. Mucus cells in the neck region of the gland secrete thin, watery mucus. Parietal cells secrete acid and intrinsic factor. Chief cells secrete pepsinogen. These paracrine cells secrete the local chemical messenger histamine. Surface epithelial cells secrete thick alkaline mucus. Click the pyloric region of the stomach to see its gastric pits and glands. In the pyloric region, gastric glands contain cells that produce gastrin and cells that produce mucus and pepsinogen. Click a G cell to see its secretion. G cells secrete the hormone gastrin. The stomach secretes gastric juice and chemical messengers that control GI activities. Stomach secretions are important for protection, digestion, intestinal absorption of vitamin B12, and control of gastric activity. Click Protection to learn more. Surface epithelial cells and mucus cells secrete a thick mucus layer that covers the gastric mucosa. Mucus protects the stomach from self-digestion by neutralizing stomach acid and inhibiting pepsin, a protein-digesting enzyme. The mucus, along with tight junctions between epithelial cells, constitutes the gastric mucosal barrier. Click the gastric pit to see an animation about this barrier. The gastric mucosal barrier provides an environment in which strong acid and digestive enzymes can break down ingested food while sparing the stomach from destruction. Click the acid character to continue. Fat-soluble compounds that diffuse through mucus and destroy the underlying epithelial cells disrupt the mucosal barrier. The only two compounds that are absorbed from the stomach, alcohol and aspirin, can break that barrier. Note that no other foods are absorbed across the stomach. Click the martini to continue. When the mucosal barrier is damaged, gastric acid and pepsin enter the stomach wall. They destroy more cells, cause release of more acid, and damage underlying layers. This damage may produce peptic ulcers. Click the epithelial cells to return to the study of protective functions of the stomach. The parietal cells of the gastric mucosa actively secrete hydrochloric acid, a strong corrosive acid that drops the pH of the stomach to 1.5 to 2. Hydrochloric acid kills most ingested bacteria, rendering chyme nearly sterile. Click Digestion to continue. In addition to its protective function, hydrochloric acid prepares stomach contents for digestion by enzymes. Drag the acid character to the spinach. Hydrochloric acid breaks down plant cell walls and connective tissue to reduce the size of food particles. Acid denatures proteins to make them more accessible to chemical digestion. Pepsin, like all protein-digesting enzymes, is produced in an inactive form called pepsinogen. If chief cells produce the active enzyme, they would self-digest. Drag the acid character to the pepsinogen. Hydrochloric acid activates pepsinogen to produce the protein-digesting enzyme pepsin by cleaving a small peptide fragment from the molecule. Drag the pepsin character to the pepsinogen. Pepsin also activates pepsinogen. Drag the pepsin character to a piece of steak. Pepsin begins the chemical digestion of protein. It cleaves protein into peptide fragments and some single amino acids. Click Intestinal absorption of vitamin B12 to continue. Vitamin B12 requires intrinsic factor to be absorbed in the intestine. Secretion of intrinsic factor is the sole essential function of the stomach. Intrinsic factor, IF, secreted by parietal cells along with acid, combines with vitamin B12 to form a complex that is absorbed in the terminal ileum. Click an intrinsic factor character to continue. Vitamin B12 is essential for maturation of red blood cells. The absence of intrinsic factor prevents absorption of B12 and leads to abnormal production of red blood cells, which causes pernicious anemia. Click Control to complete your study of stomach functions. Peptides stimulate release of the hormone gastrin. Click the gastric pit to see the actions of gastrin and hydrochloric acid. Gastrin is the most important factor for stimulating acid secretion. Gastrin stimulates parietal cells to secrete hydrochloric acid and stimulates paracrine cells to secrete histamine. Histamine also stimulates parietal cells to release hydrochloric acid, reinforcing the action of gastrin. Like all the GI hormones, gastrin has many functions. In general, gastrin promotes gastric motility and secretion and exerts a trophic effect on the stomach. Gastrin acts on parietal, paracrine, and chief cells of the stomach to increase gastric secretion. Gastrin is important for moving the contents of the previous meal out of the terminal ileum as a new meal enters the stomach and for inducing a mass movement. You may click any of the stomach functions at the top of this page to see them again or click to continue. Create a table that lists the functions of gastric secretions by dragging each phrase to its correct position. Click Begin to start. On this page, we will build a table that illustrates the regulation of gastric secretion. Think about what you have learned about neural reflexes and GI hormones and answer each question as it comes up. You must answer the question correctly to proceed. During the cephalic phase of control, stimuli like the thought, sight, smell, or taste of food elicit a response of the stomach. Will nerves or a hormone mediate the response? Will the neural response involve extrinsic nerves, a long reflex, or only the enteric nervous system, a short reflex? Long neural reflexes increase gastric secretion of hydrochloric acid and pepsinogen directly by stimulating two types of stomach cells. Which two? Select the cells that produce each secretion. Select the cells that produce each secretion. Select the cells that produce each secretion. Long neural reflexes increase gastric secretion of hydrochloric acid and pepsinogen indirectly by stimulating release of gastrin. Which cells release gastrin? Gastrin stimulates release of histamine, which reinforces acid secretion. Which cells release histamine? What term describes locally acting chemical messengers? Will gastric secretion of hydrochloric acid and pepsinogen increase or decrease as the stomach prepares to receive a meal? During the gastric phase of control, peptides in the stomach and distension of the stomach wall elicit a response. Will nerves, a hormone, or both mediate the response? Which hormone mediates this response? Note that we use the term neural reflexes when the neural mechanism is not fully understood, or when both long and short reflexes are involved. Remember that long reflexes to the stomach involve the vagus nerve, and that gastrin stimulates parietal cells both directly and indirectly by causing release of histamine. Will gastric secretion increase or decrease as the stomach begins to work? When a meal enters the duodenum, the intestinal phase of control begins. Distension, fat, acid, peptide fragments, and hypertonicity elicit a response. Will the response be carried out by nerves or a hormone? Think carefully about what the intestinal hormones do and what the appropriate stimulus for release of each might be. Hormones are involved, but that's not all. Which two hormones mediate this response? Fat causes release of CCK, and CCK affects gastric secretion. HCl causes release of secretin, and secretin affects gastric secretion. Will gastric motility increase or decrease as chyme fills the duodenum? Think about the function of the small intestine. This is the enterogastric reflex we first studied in the topic motility. The reflex dampens both gastric secretion and motility until chyme is neutralized and rendered isosmotic. Only then can maximal digestion and absorption occur. Excessive acid secretion in the stomach exerts negative feedback on gastric secretion. Will secretion increase or decrease when the pH of the stomach falls below 3? In addition to the regulation of digestion that is governed by food and the thought of food, the autonomic nervous system can exert control over the operation. What will happen to digestive activity when the sympathetic nerves are activated? What will happen to digestive activity when the parasympathetic nerves are activated? Hydrochloric acid and pepsinogen are actively secreted when the stomach prepares for and contains a meal. Once the meal has started to enter the intestine, feedback adjusts gastric secretion to maximize intestinal digestion and absorption. Most digestion and absorption occurs in the small intestine. The essential secretions for digestion come from the pancreas and liver, not from the small intestine. We will study secretions of these accessory glands first, beginning with the pancreas. The exocrine pancreas secretes a juice with two components that are regulated separately: enzymes to digest all major foodstuffs and a bicarbonate solution. CCK stimulates secretion of pancreatic enzymes, and secretin stimulates secretion of bicarbonate solution. Click the pancreas to begin. Pancreatic juice is important for digestion and protection. Click Digestion to learn more. Pancreatic juice is produced in structures called acini and carried through a network of ducts to enter the duodenum through the hepatopancreatic sphincter. Click the acinar cells. Acinar cells secrete digestive enzymes for all major foodstuffs. They include proteases to digest protein, pancreatic amylase to digest starch, and pancreatic lipase to digest fat. Click the duodenum. The major pancreatic proteases are trypsinogen, chymotrypsinogen, and procarboxypeptidase. Like pepsin, they are secreted in an inactive form. Click the intestinal cell. Enterokinase, an enzyme embedded in the intestinal cell membranes, activates trypsinogen. Drag trypsinogen to enterokinase. Trypsin activates more trypsinogen. This process is similar to activation of pepsinogen in the stomach, first by hydrochloric acid, then by pepsin. Trypsin also activates the other proteases. Click enterokinase to repeat the animation, or click the duodenum to continue. Carbohydrates and protein are partially digested prior to arriving in the small intestine. Fat is digested primarily by pancreatic lipase in the small intestine. Therefore, if the pancreas fails to secrete adequate amounts of digestive enzymes, the major disruption in digestion is the failure to digest fat. Click Protection to continue. Duct cells secrete a watery solution containing bicarbonate. Bicarbonate neutralizes acidic chyme in the duodenum. A neutral to slightly alkaline intestinal environment is essential for maximal activity of pancreatic enzymes. Remember that the pancreas is a mixed gland with both exocrine and endocrine secretions. The endocrine pancreas secretes hormones including insulin and glucagon that regulate metabolic activities of the absorptive and post-absorptive states respectively. The major digestive function of the liver is the production of bile. Bile is produced continuously. When the hepatopancreatic sphincter is closed, bile goes to the gallbladder where it is stored and concentrated. Bile has two components that are regulated separately: organic compounds that are essential to emulsify fat, and a bicarbonate solution. Recycled bile salts in portal blood regulate secretion of bile, and secretin stimulates secretion of bicarbonate solution. Click the liver to begin. Bile is important for digestion and absorption of fat, and protection. Click digestion and absorption of fat to learn more. The organic portion of bile is a mixture that includes bile salts, lecithin, cholesterol, and bilirubin. Bile does not digest fat. Both bile salts, which are derived from cholesterol, and lecithin emulsify fat. They keep fat droplets from coalescing, thus promoting enzymatic breakdown and absorption of fat. We will study digestion and absorption of fat in detail in the next topic. Cholesterol and bilirubin, a breakdown product of heme, are eliminated. Click the gallbladder to observe circulation of bile salts. Bile salts are recycled. Bile enters the duodenum during a meal and travels through the small intestine. Bile salts are then actively reabsorbed in the terminal ileum and returned to the liver in the hepatic portal blood. In the liver, bile salts stimulate secretion of bile. This pathway is called the Enterohepatic Circulation. Bile salts may circulate through this pathway several times during digestion of a meal. Click Protection to continue. Cells that line the bile ducts secrete a watery solution containing bicarbonate. This solution is identical to pancreatic bicarbonate solution and neutralizes acidic chyme in the duodenum. Secretin promotes secretion of both. Cells that line the bile ducts secrete a watery solution containing bicarbonate. This solution is identical to pancreatic bicarbonate solution and neutralizes acidic chyme in the duodenum. Secretin promotes secretion of both. The small intestine secretes a watery mucus solution and hormones that control GI activities. Small intestine secretions are important for protection, digestion, and control. Click Protection to learn more. The intestinal mucosa is specialized for absorption. Mucus-secreting goblet cells are plentiful. Mucus protects the intestinal mucosa from digestion by acid and proteases and lubricates the contents for ease of movement. Click the portion of the small intestine that is at greatest risk for acid damage. The submucosa of the duodenum contains mucus-secreting duodenal glands that secrete alkaline mucus. Click Digestion to continue. Cells of the crypts secrete the water and electrolytes that combine with mucus to form intestinal juice. Water provides an environment for digestive reactions and adjusts osmolarity. Click the jejunum. Cells of the small intestine do not secrete any digestive enzymes into the lumen, but they do produce digestive enzymes. These enzymes, called brush border enzymes, are bound to microvilli cell membranes. We have already encountered the brush border enzyme enterokinase, which activates trypsinogen. Click Control to continue. In general, secretin and CCK promote intestinal activities and inhibit activities of the stomach. Contents in chyme stimulate secretion of the intestinal hormones, and both hormones have trophic effects on the pancreas. Drag a droplet of acid to an enteroendocrine cell. Acid in the duodenum stimulates secretion of secretin. Secretin stimulates duct cells of the pancreas and the liver to release bicarbonate solution. Because of its actions, secretin is called nature's antacid. Drag a droplet of fat to an enteroendocrine cell. On this page, we will create a table that illustrates the regulation of secretions that enter the small intestine. Think about what you've learned about neural reflexes and GI hormones and answer each question as it comes up. You must answer the question correctly to proceed. Since most intestinal activity occurs as a meal enters the duodenum, we will focus on the intestinal phase of control. During the intestinal phase of control, acid in the duodenum stimulates release of an intestinal hormone. Which hormone? Which hormone is nature's antacid? Secretin acts on the pancreas and liver. Which cells does it stimulate? Which secretion enters the small intestine in response to secretin? Will secretion of bicarbonate increase or decrease during the intestinal phase? Fat, and to a lesser degree peptides, in the duodenum stimulate release of an intestinal hormone. Which hormone? When CCK acts on the pancreas, which cells does it stimulate? Which secretion enters the small intestine in response to CCK? Will secretion of digestive enzymes increase or decrease during the intestinal phase? CCK causes bile to enter the small intestine by action on another organ. Which organ? Distension of the small intestine and acidic or hypertonic chyme affect mucus and water secretion of the small intestine. Will nerves or a hormone mediate the response? Stretch of smooth muscle often elicits a neural response. Will secretion of intestinal juice increase or decrease? In addition to the regulation of digestion that is governed by food and the thought of food, the ANS can exert control over the operation. What will happen to digestive activity when the sympathetic nerves are activated? What will happen to digestive activity when the parasympathetic nerves are activated? Small increases in pancreatic and hepatic secretions may occur during the cephalic and gastric phases of control, but most activity in the small intestine occurs as it digests and absorbs a meal. Secretion of bicarbonate solutions, digestive enzymes, bile and intestinal juices increase during the intestinal phase of control. The large intestine secretes an alkaline mucus solution containing bicarbonate and potassium. Its sole function is protection. The alkaline mucus solution protects the intestine wall from damage by acids released by resident bacteria. Mucus eases the passage of feces and protects the wall from mechanical damage. Acid and mechanical stimulation, mediated by both long and short reflexes, increase the secretion of alkaline mucus. Here's a summary of what we've covered. Mucus protects the entire GI tract and eases passage of its contents. The stomach and pancreas produce digestive enzymes. The stomach and small intestines secrete hormones that regulate function of the entire tract. To test your knowledge, click the quiz button to go to the self-quiz. To access cross-references for this topic in your Benjamin Cummings textbook, click here. To test your knowledge, click the quiz button to go to the self-quiz.